Okay, before we begin, are there any questions? I'm going to take that as a no. Let me share my screen. So today is May 24th, and we should be starting principles of disease. I don't think we're finished with microbial genetics yet. Is that right? Did we finish genetics?
We did.
We did. All right. Yeah, I did. I got that marked right there. Yeah. Open up the other chapter then. Hopefully I have it here. Yep, I do. So we're just a little bit behind schedule. We were supposed to have started chapter 14 last time, but we have a day and a half on principles of disease, chapter 14. So I think we'll do all right. Please note there is no lab all week, meaning both Tuesday and Thursday. I will be logging in at 6:30 to answer any questions concerning the lab or any other questions. But after 6:45, if there's no further questions, I am going to log off. Any questions about that?
I have a question, Dr. Volpe.
Uh-huh.
Do you need to know if we have already identified our unknown?
I don't need to know that. You can put it in your unknown notes.
Okay.
Yeah, you don't need to notify me of that.
Okay, thank you.
All right. Let me remind you that you have a quiz this week, quiz 5. And you can start it, I believe, Tuesday at 8 p.m. It must be taken by Wednesday at 8 p.m. It covers chapters 8 and lab 9. So this quiz will be over less material than the previous quizzes. So just chapter 8 and lab 9.
Sorry, I was wondering, like, since there is no lab today, do you think that quiz can be opened a little bit earlier?
No, we've decided in the biology department to make the quizzes only open for 24 hours. And I don't want to change the schedule because hopefully you guys have gotten used to it from 8 p.m. to 8 p.m.
Okay, thank you.
Okay. I used to have it 11:59 p.m. to 11:59 p.m. But students, several of them requested I have it earlier because of work schedules. And most people don't take the quiz after 11:59 p.m. Tuesday night anyways. They would wait until Wednesday night to take it. All right. There's no further questions. Let's get started. Can you guys hear me all right? I've got the headphone on now.
Yes.
Assuming you can hear me, so. Oh, look at that. I even had a mark there to remind me to start here. So chapter 14, principles of epidemiology, and I can't touch on this. Why is that? Oh well. Principles of disease and epidemiology. The major goals and the rough outline. Know the terms. I've only got 3 or 4 terms listed here. However, this chapter is a lot of terms, and if you know all the terms to this chapter, you will know about half of the chapter. Any questions about that? Why can't I select that? Something's wrong with my PowerPoint for some reason. Understand the relationship of the normal microbiota and its host. The two are together and understand that relationship. Understand the stages of disease development, and understand what nosocomial infections are and how to control them. Any questions about what we're doing? All right, the first terms I want to talk about are pathology, the study of disease, etiology, the study of the cause of a disease. For example, the etiological agent of anthrax is Bacillus anthracis. And this was the first bacteria that was shown to cause a disease. Pathogenesis is the development of a disease.
So for pathogenesis, is that like when scientists try to figure out how a new disease came about to be?
Not really. It's more about when a patient gets the disease and then the development of the disease.
Okay, kind of like how the
Talking about
flu
how
progresses.
the disease evolved, like COVID-19. We'll talk a little bit about that later in the term, but how that came about, that isn't really pathogenesis. Pathogenesis will be the development of a patient coming down with COVID-19 symptoms, signs and symptoms.
Okay, thank you.
Sure. Infection is the colonization of the body by pathogens, or the presence of a microbe where it is not normally found. For example, E. coli is most of the time living in harmony with us. And if it's in your gut, nothing's wrong. But if the E. coli is growing in your ear, causing an ear infection, or in your lungs, causing a lung infection, that would be an infection where the microbe is not normally found. A disease is an abnormal state in which the body is not functioning normally. You should understand that only a few microorganisms are pathogenic. Most microorganisms are not pathogenic. Some can even benefit the host. And some that benefit the host we call the normal microbiota. Now, not all of the normal microbiota benefits the host. The normal microbiota is a large community of microbes found in association with all animals and plants. These communities are known as the normal microbiota, the microbes that permanently colonize a host. For humans, we would call it the human normal microbiota. And it would be slightly different than, for example, the cat normal microbiota. For example, Bartonella henselae is a normal microbe living in and on the cat. And I don't know exactly where that Bartonella henselae is on the cat, but I know it's in the claws and in the mouth. Bartonella henselae does not normally grow on humans. And when it's in a human or on a human, it usually causes disease. Any questions about any of that? So something that's a normal microbiota for one animal can cause disease in another. Another example, a good one, is salmonella is in the gut of many birds. So it's a part of their normal microbiota. But for humans, if it's in our gut, it causes disease. The normal microbiota are only found in certain regions of the body. And disruption of the normal microbiota and the host balance appears to be associated with diseases and impaired immune function. For example, when a patient first takes antibiotics orally, it does kill off, hopefully, the pathogen that they're prescribing the antibiotics to. but it also kills off a large number of your normal microbiota of your gut. And that can cause disease, causing the patient to either have diarrhea or become constipated. And I'm sure that's happened to many of you in the past. In fact, that's happened to me numerous times when I take antibiotics. Sometimes I get diarrhea, and it usually gets better after a few days. Any question about that? All right, the locations of the normal microbiota in the human body. It's found obviously on the skin, on all of the mucous membranes open to the outside, which I think are all of the mucous membranes, like the eyes, the mouth, the nose, the upper respiratory system, like the nose and the throat. The normal microbiota is not normally found in the lungs. Okay, so the lower respiratory system usually does not have the normal microbiota. The gastrointestinal system has normal microbiota from the mouth all the way down to the anus. The urinary system, what the heck am I trying to say? Normally only has normal microbes growing in the lower urinary and reproductive systems, like the urethra has a normal microbiota. Okay. Any questions about that? But up higher, like the ureters and the kidneys, do not normally have microbes. And if the microbe were to grow in the urethra and then spread to the bladder, then spread to the ureter, and then climb up the ureter to the kidneys, that usually causes disease. Okay? So anything past the bladder, as far as I know, is associated with disease. And we'll talk about the bladder in a minute. The normal microbiota is not normally found in the blood, the cerebrospinal fluid, muscles, nervous tissue, bone, anything internal. If it's not open to the outside, there's no normal microbiota normally there. And if we have bacteria in the blood, that would be an infection. Any questions about any of that? We can have a transient microbiota that may be present for days or months, but unlike the normal microbiota, which when it colonizes the host, it generally stays with the host for the host's lifetime. Like for example, when a baby is born, they normally do not have Staphylococcus epidermidis until birth. Any questions about that? But after birth and actually during birth, they pick up Staphylococcus epidermidis. And then it stays with the baby generally for the entire life. And that's the normal microbiota. Transient microbiota is much shorter. For example, my aunt had Staphylococcus aureus skin infection, and she was taking antibiotics for it. And of course, we went to visit her, me and my mother, and she insisted on hugging me. And I noticed the next time I tested my skin in the lab, I had Staphylococcus aureus for the first time on my skin. And I said, geez, I must have picked that up from my aunt when she hugged me, because I never had it before. And it was a transient, meaning the next time I tested my skin and all further times I tested my skin, I never saw Staphylococcus aureus on my skin any further. So it stayed on my skin for less than 3 months. And I'm not sure how long it was there because I probably just tested it one term. And about 3 months later, I tested it again. Any questions about any of that? So fortunately for me, it didn't cause any disease like it did for my aunt. And I didn't need to get rid of it with antibiotics. That it was only transient and it went away on its own, probably because it was competing with the other microbes on my skin and it just lost out. When we're talking about the normal microbiota and the host, the two live together. When we're talking about humans, humans have about 1 times 10 to the 13th human body cells. And humans have about 1 times 10 to the 14th bacteria cells living on and in them. That begs to answer the question, are we more microbe than we are human? Any questions about that? Don't let that freak you out too much. But yeah, we have more microbes than we have human cells. Of course, the microbes are smaller than human cells too. Here are some of the normal microbiota. The bacteria, the orange cocci in the nasal epithelium. Bacteria in the stomach, it's the, gosh, what are those? Spirochetes? Spirals and spirochetes in the stomach. That is not bacteria, that's the lining of the stomach. And then bacteria in the large intestine, you can see rods here, and then coccobacillus there, probably a cocci there. And that right there is material that somebody's eaten. Some unusual places that we didn't know initially humans had microbes in these locations, but we discovered it around 2014, maybe a little earlier. The urine in females, as you know, has bacteria in it because of the urethra. Well, it turns out at least females have bacteria in the bladder. And the study did not look at male bladders, so I don't know the question, but apparently the female bladder has a normal microbiota in the bladder. Any questions about that? Mother's milk has bacteria in it, and I think initially people just assumed that it came from the outside of the nipple, which we knew had bacteria on it. But when they look at it and they looked at the breast tissue, they find that there's normal microbiota inside the breast tissue. So mother's milk, even inside the mother, has normal microbiota. Any question about any of that? And this one, I don't know why this is a recent surprise. They found that the female placenta has a normal microbiota. And why, I'm not sure why that's a surprise, is that the female reproductive system is open to the outside. So the placenta getting microbes is not a surprise to me. But when a female is pregnant and has a placenta, apparently there are microbes that colonize this area and make a normal microbiota. And then this normal microbiota goes away when the baby is born or the mother miscarriages and there's no more placenta. All right, there is a normal relationship between the normal microbiota and the host, and the normal microbiota can help the host by microbial antagonism. meaning the normal microbiota can compete with the bacteria that can cause disease, the pathogens. They can compete by preventing the growth of the pathogen. For example, on my skin, there's lots of normal microbiota there. If I were to get one human pathogen, it may not have room to grow on the skin, meaning there may be bacteria already there and there won't be room for the pathogen to get established and then grow. Any questions about that? That's occupying a niche that the pathogen might occupy. It can compete for resources. Even if it does get on the skin, there may be normal microbiota around it, and then the pathogen will have to compete for resources, and it may not get enough to grow, in which case the normal microbiota will prevent the disease. The normal microbiota can produce acid that will deteriorate the environment so that the pathogen would not be able to grow. An example of that is, well, actually there's two examples. The urethra has normal microbiota on it that lowers the pH, which prevents some pathogens from growing in the urethra. Also, our skin has normal microbiota, which decreases the skin pH further. Let me see, the skin secretes sebum, which is slightly acidic, but the normal microbiota can metabolize the sebum and make it even more acidic. So the skin is slightly acidic. It's not strongly acidic like your stomach, but it is slightly acidic from your normal microbiota, as well as the sebum, which is the lubricant that your skin makes. The normal microbiota can also produce bacteriocins. This is usually a protein which acts sort of like an antibiotic. However, a bacteriocin is much more specific and narrow in its field of action. For example, a bacteriocin produced by E. coli would prevent the growth of salmonella and shigella in the intestine, if that's where the E. coli is, but it would not prevent other bacteria that are less related to E. coli. And I'm trying to think of something in your gut, like, what is it? Fecalis. You know which bacteria I'm trying to say? Drawing a blank there. It's one that's very common in your gut, something like Fecalis. And the E. coli bacteriocin would not inhibit that. So it would inhibit some bacteria that could, if it got into your intestine, cause disease, but it doesn't interfere with other bacteria which are normal in your intestine. So it would not inhibit them. And I've often wondered why we don't use bacteriocins as an antibiotic, because they're very specific in their action. It'd be much more specific than, let's see, you'd have to take a gram-negative to like cure salmonella if it was in your intestine. So you have to have an antibiotic that inhibits at least gram-negatives to kill off the salmonella. And that would kill off most of the bacteria in your gut, because most of them are gram-negatives. If you were to take a bacteriocin that were to prevent salmonella and shigella, it would kill off the salmonella, and hopefully you wouldn't have any shigella in your gut, but if you had any, it would kill that off. but it wouldn't kill off all the other gram negatives in your gut. And so this would be actually a very good antibiotic. And I did read an article many years ago that they were experimenting with using bacteriocins as antibiotics. I haven't seen anything further, but at least they were experimenting with bacteriocins. And like I said, it would be a very good antibiotic-like drug. Any question about any of that? We can also take microbes as probiotics, the consumption of live microbes that are intended to exert a beneficial effect. For example, the microbes in yogurt can be taken as a probiotic, excuse me, and not only can they help the microbes in your gut, but the microbes in yogurt can actually decrease the risk of a bladder infection in women. And for some reason, actually I do know why, but I'm not going to get into it. When you eat yogurt that has live cultures, the bacteria from the yogurt can colonize the urethra, which would then act as a normal microbiota in the urethra, and because of antagonism, prevent the growth of the pathogen. Any question about any of that? All right, so let's talk a little bit more about the relationship of the normal microbiota and the host. The normal microbiota is living in symbiosis with the host. Symbiosis is simply when two different organisms or populations are living together. How many of you have cats or dogs?
Yep.
Living in your home. Okay, then you're living in symbiosis with the cat and the dog. Two different organisms living together. The normal microbiota and the host live together in symbiosis. And there's three different types of symbiotic relationships. There's commensalism, where one organism benefits and the other is unaffected. Some of the normal microbiota are commensalistic, meaning they practice commensalism. They're getting a home. They may even be getting food and resources from us, but they're not really benefiting us. I can think of one example. It's hard coming up with an example that actually gives us no benefit, because usually there's a little bit of slight benefit, and that is the normal microbiota can compete with the human pathogen, and that would be giving us a little benefit. And if it prevented disease, it'd be a pretty big benefit. But I do know one commensalistic, it's actually a microbe. It's a microscopic nematode, a worm that you can find in your eye. It lives in the fluid of your eyeball. And I don't know what it eats, probably bacteria that falls into your eye, because it doesn't do us any damage. And I think we have, on average, humans have about one per eye. So I guess every human would have two, because we have two eyes. Assuming you have two eyes. Any question about that? It doesn't do us any damage. It doesn't eat any of our eyeball. It doesn't really do us any good, but we're giving it a home. We're not feeding it, as far as I know. Like I said, I don't know what it's eating, but I'm assuming it's eating bacteria that fall into your eye. A lot of the normal microbiota have a mutualistic symbiotic relationship. Mutualism is where both organisms benefit. Like the E. coli living in your gut benefits because it gets a home and then we feed it. When we eat food, the E. coli can digest it and then grow and reproduce in your gut. So it's benefiting. It's benefiting us because the E. coli in our gut helps us digest our food. There are some other slight benefits, like if the E. coli produces bacteriocin, it can compete with normal microbes and prevent the growth of, not normal microbes, it can prevent the growth of human pathogens. The Staphylococcus epidermidis on our skin largely acts as a commensalistic organism, meaning it's not giving us any direct benefit, but it is competing with a human pathogen. And so the human pathogen may not get established on your skin. And so most of the time it's commensalistic, but at least some of the time it's even mutualistic, where it prevents the pathogen from getting established. Another thing it does is it changes the skin, making it more acidic, and that prevents the growth of some pathogens, so that would be also mutualistic. Anyways, the normal microbiota are normally commensalistic or mutualistic. Now, the third relationship is parasitism, where one organism benefits at the expense of the other. For example, those of you who have cats, you provide the cat a home, you feed the cat, and you buy the food and the kitty litter with your own money that you work hard for. And then the cat doesn't give you anything in return. So that is parasitism. Any questions there?
I think you have the wrong idea about cats.
So what's the benefit the cat gives you? You don't eat the cat.
They're good friends.
They're a friend. And how does that benefit you? Come on, you can say. You can say that the cat benefits your health because it pleases you, and that increases your immune system, and that way you're more likely to survive if you were ever to get an infection. And there are times when your cat can give you some benefit. Okay. I was just being a little bit facetious and trying to be a little bit hilarious when I was saying that cats are parasitic. I don't believe the cats are parasitic. A parasitic organism would be a human pathogen that would be something like Staphylococcus aureus when it's growing on your skin and then it causes skin disease. Okay, that would be parasitism, where one organism benefits, it's getting a home, it's getting food from you, and then it's causing you disease. Okay. And obviously a cat doesn't cause you disease. All right. The normal microbiota normally is commensalistic or mutualistic to us, but occasionally it can be opportunistic. An opportunistic microorganism is usually a normal microbiota that can cause disease under certain circumstances. Now, the normal microbiota does not normally cause disease in a healthy individual. But on certain individuals, like somebody who has an immune compromised system, it can cause disease. Like a patient who has AIDS, they don't have an immune system, and the normal microbiota can grow in regions where it shouldn't be growing and then cause disease. An example would be E. coli, which normally is found in the gut. And when it's in your gut, it's a part of your normal microbiota. But if it's growing in your ear or in your lungs, it can cause disease. Staphylococcus aureus actually is, at least in some people, no part of your normal microbiota in your nose and your throat. But it can cause disease on your skin. And I have to state that it doesn't always cause disease on your skin. About 20% of Americans have Staphylococcus aureus growing on their skin. And most of the time, it does not cause disease. However, if you're an individual who frequently gets skin boils, you might have yourself tested to see if you have Staphylococcus aureus growing on your skin. Because people who get skin boils actually frequently have Staphylococcus aureus on their skin, and every once in a while it causes a little infection that we call a skin boil. It can cause more serious disease if it ever grows beyond that. All right, the etiology of an infectious disease. The first case of a bacteria that was shown to cause disease was discovered by Dr. Koch when he found that Bacillus anthracis causes the disease anthrax. And when he published his findings, one of the things he did was he published the steps he went through to show that Bacillus anthracis causes the disease anthrax. And let's go through this a second time. We will go through this a third time. So obviously this is an important part of microbiology because we're covering it in 3 different places. And it's known as Koch's postulates. The steps you use to show or prove that a given agent causes an infectious disease. First, you have to isolate the pathogen from a sick individual. And then you grow up the pathogen in the laboratory. And then you isolate the pathogen, grow it up as a pure culture. And usually how you do that is you take a single isolated colony and then streak it out on another plate to get isolated colonies. And then when it's gone through 2 colony isolation events, set up your culture. And that's likely going to be a pure culture, greater than 95% chance that that'll be a pure culture. And then you take that culture and grow it up, and then you inject it into a healthy animal. And then you look to see if that animal comes down with disease. Why the bunny is tilted on its side. It's a sick bunny. It's got disease. Ah, poor bunny. And that disease has to be the same as the first disease, meaning the same signs and symptoms, or at least similar signs and symptoms. And then Dr. Koch said you had to go one step further. You need to isolate that organism from meaning the organism causing the disease from the host, the patient, or in this case, the sick bunny. Any questions about Koch's postulates? Oops. No questions, I'm going to move on. Sorry, didn't mean to go there. All right. There are exceptions to Koch's postulates. What do you do with a microbe that has unique culture requirements and it cannot be grown on artificial media? How can you prove that this bacteria causes disease? Actually, sorry? Actually, I know of two diseases where we've been trying to grow them for over 100 years. And gosh, this has got to be over 120 years now. They have never been successful in growing syphilis in the lab. Another one is Bartonella henselae, which causes cat scratch fever. They've never been able to get that to grow on a tissue culture plate. We can only get it to grow in another animal. Okay. So what did we do to show that something like syphilis or Bartonella henselae causes the human disease? Because we can't use Koch's postulate to show that this organism is causing the human disease. So what did we do?
Could you expose another organism to the disease and see if that organism develops similar symptoms?
Yeah, you could do that. In fact, they have done that with syphilis. It grows at least in rabbits. Okay. And it does cause disease in them. I'm not sure what the disease is, but so that's one thing you can do. Well, what about a disease, something like AIDS, that it only grows in humans, causing disease only in humans, HIV? How do we know that HIV causes AIDS if we can't use Koch's postulates? And trust me, we've never injected HIV in a patient to see if it then causes AIDS, because AIDS is just a too horrible disease. It would be unethical as well as immoral to inject a patient with HIV. So what did we do to show that HIV causes AIDS? Wasn't Koch's postulates. Nobody has any idea? You did vast epidemiological studies. You followed large number of people over time, and some of those people picked up HIV on their own. And then you follow these people who became HIV positive over time. And we noticed that they tended to get AIDS. Okay. And then you also follow the people who stayed HIV negative, and they do not get the disease AIDS. So the vast epidemiological studies over time can show a given microorganism is capable of causing a given disease. So those are actually two different ways that we can show that a given organism causes disease: Koch's postulates and then vast epidemiological studies. Any question about that? What do you do if multiple organisms cause the same disease signs and symptoms? For example, the human cold can cause, let's make this a human here, can cause us to get sick, and we can isolate that and take that virus and inject it into another patient. And the other patient can come down with the cold. However, we can take another virus and inject it into the patient, and they can come down with a cold. There's many different species of viruses that cause the human cold. So Koch's postulate really doesn't work. Because there's more than one organism that can cause the human cold. So Koch's postulate doesn't work. You can use vast epidemiological studies to test if a given organism causes the cold. For the virus, they've also injected it into people and found that they developed the cold. And that's pretty strong evidence that that's not exactly Koch's postulate, but it's a modification of Koch's postulate that virus can cause a human cold. And then some species can cause different disease conditions, different disease signs and symptoms. For example, is it salmonella? Yeah, salmonellosis can cause, salmonella can cause salmonellosis, and it can cause, is it typhoid fever? I'm drawing a blank here. It's one of those fevers. I can go online and check. We have time. I think we have time. Yep, salmonella. All right. So the question is, what do you do in those cases? Like I said, Koch's postulate doesn't work directly, but you can use vast epidemiological studies. You could also give a patient or several patients this organism. And even though it may cause two different signs and symptoms, you would state that it does cause typhoid fever because some of the patients come down with typhoid fever. And then other patients come down with salmonellosis. All right, any questions about any of this? Exceptions to Koch's postulates? A symptom is a change in the body function that is experienced by a person as a result of disease. But it is a subjective, phenotype symptom, meaning it is not apparent to another observer. Somebody else cannot measure a symptom. For example, when a patient gets a headache, they're the only one who knows they have a headache. Nobody else can measure that, oh, yeah, this person has a headache. So a headache is a symptom. A sign, on the other hand, is a change in the body that can be measured or observed as a result of disease. An example is the temperature of 38.5 degrees Celsius. You can put a thermometer in somebody and then measure it and say, yep, you've got a temperature. So a temperature of 38.5 would be a sign. Other people can measure it. Any question about any of that? Now, this is a textbook definition of a sign and symptom. You'll find that in the general public, these two terms are used interchangeably. And in fact, even in the clinic, even with the doctors and other clinicians, they tend to use sign and symptom interchangeably. And so I won't test you, at least I don't think I'm going to test you on what is a sign and what is a symptom, because we will use them interchangeably, at least some of the time. The syndrome is a specific group of signs and symptoms that characterize a disease, such as the AIDS syndrome, the group of signs and symptoms that characterize the disease AIDS. A communicable disease is a disease that is spread from one host to another. Examples of a communicable disease are measles, TB, tuberculosis, and chickenpox. A contagious disease is a disease that easily spreads from one host to another. What's an example of a contagious disease?
The flu.
The flu is a contagious disease. What's another example?
COVID.
COVID-19 is another example. What's an example of a communicable disease but is not contagious? Meaning it does spread, but it doesn't spread easily. Nobody has an idea? Actually, tuberculosis. Somebody's going to say something?
Would HIV count?
Yeah, in a way, it is true that you only need one exposure, but HIV does not easily spread from one host to another. And I don't remember how many exposures you need on average to become infected with HIV, but it does not spread easily, at least in a healthy individual. The trouble is if you have a pathogen that is asymptomatic, it can damage your mucosa in the reproductive system, and then you'd be much more likely to get HIV. In fact, most people, assuming that they have an intact mucous membrane, they cannot get HIV. The trouble is about 70% of the American population have, well, I'm going to call it a human pathogen. You could call it a normal microbiota as well. They have a bacteria, or yes, a bacteria that damages the human mucosa. Assuming that you've been reproductively active, about 70% of Americans have this bacteria. And most of the time, at least in young, healthy individuals, you're asymptomatic. But this bacteria does damage your mucosa. And if you have this bacteria, you have a damaged mucosa, and then you can get HIV infection. And about 70% of Americans can get an HIV infection because they have a damaged mucosa. Any question about any of that? A better example of a communicable disease that's not contagious would be tuberculosis. Tuberculosis can spread while it's communicable, but it does not spread easily. Another one is leprosy. Mycobacterium leprae can spread, but it does not spread easily. You need long-term exposure for both TB and leprosy before you will come down with the disease. Any question about any of that? And then a contagious disease is always communicable. And an example would be something that spreads very easily, like COVID-19 or a virus causing the human cold. Contagious disease. A non-communicable disease is one that does not spread from one host to another. For example, you can get tetanus by stepping on a nail, but once you get tetanus, you are unlikely to spread it to anybody else. So it is a non-communicable disease. The other thing would be, what does that disease cause? When you eat canned food, botulism. It is a non-communicable disease. You can get it by eating tainted canned food or maybe some other tainted food. But once you get it, you're unlikely to spread it to another patient. Okay, so it's a non-communicable disease. And I can only think of one example where tetanus or botulism can spread from one patient to another. And at least theoretically, these diseases could spread in the case of cannibalism. But other than that, they're non-communicable. So if you do practice cannibalism, you should make sure that your human is cooked very well. Any questions? I'm just trying to be a little hilarious there. I don't mean to offend anyone. Obviously, I don't want you eating humans. And most people don't, and I shouldn't even talk about the subject. Occurrence of disease. To understand the full scope of a disease, we need to understand the details about its occurrence. We need to understand the old and the new cases. The incidence is the fraction of the population that contracts a disease during a specific time. The incidence only is counting the new cases only. Like if you were to go on the news today and find out how many people came down with COVID-19 in the United States today, that would be the incidence for that day, the new cases only. The prevalence is the fraction of a population having a specific disease at any given time. For example, normally they talk about the prevalence of the number of people in the United States that have come down with COVID-19. And the last I heard, this was over, no, that's the prevalence of people who died from COVID-19. I think it was 800,000 people in the United States who have died of COVID-19. And usually that's stated as the prevalence. In AIDS in 2007, we could say in 2007, the incidence of AIDS was 56,300 cases in the United States, meaning new cases in 2007. The prevalence, though, was over 1 million. People had AIDS in the United States. Any question about any of that? A sporadic disease is a disease that occurs only occasionally in a population. For example, typhoid fever happens sporadically in the United States. An endemic disease is a disease that is constantly present in a population. Like the common cold is endemic to the human population. I'm not aware of any population of humans that do not have the common cold. Malaria is endemic to the tropics. Anywhere you go in the tropics, you can find malaria, at least in the world. An epidemic is a disease acquired by many hosts in a specific given period of time. Like every once in a while, we have an epidemic of influenza in, I don't know, the United States or North America. And that we call an epidemic. What you have specifically to separate epidemic from a normal case of the flu is an epidemic has to be significantly higher than other cases of the flu. So most of the years we have just a normal occurrence of influenza, usually in the wintertime or near the wintertime, like in the fall and the early spring. An epidemic, though, is when we have significantly higher cases. of the flu. And with the flu, I can tell you that on average, we get an epidemic of the flu about every 10 years. And we'll talk a little bit more about that when we're talking about the viruses causing the flu later in the term. A pandemic is a worldwide occurrence of an epidemic. For example, when COVID-19 first hit the world, it was only in China. So the only continent we found it on was China. And at that point, COVID-19 was an epidemic because it was significantly higher than normal, because the normal was 0 cases when we first had COVID-19. And then it spread from China and went, I think, South Korea next and Japan next, but still an epidemic because it's only on one continent. But then when it goes beyond that to the United States and Europe, it then became pandemic, a worldwide epidemic of COVID-19. Usually for very contagious diseases, a disease that's epidemic will become pandemic. And they tried controlling COVID-19, and because of asymptomatic cases, it spread beyond China. Another useful way of defining the scope of a disease is to define it in terms of its severity or its duration. An acute disease is a disease that develops rapidly. The signs and symptoms come on quickly, but the disease is short-lived. An example of an acute disease is the human cold. It hits the patient, they quickly develop signs and symptoms, the disease develops rapidly, and then it goes away quickly. So the human cold is short-lived, and we can call it an example of an acute disease. A chronic disease, on the other hand, is a disease that develops slowly. It persists for long periods of time. And usually over time, the chronic disease gets worse. An example of a chronic disease is tennis elbow, where somebody who's constantly playing tennis, and as they play tennis, it does damage to your elbow. And the more you play the tennis, the worse it'll get. And so with time, the chronic disease just gets worse and worse. Any questions about any of that? An example of a microbe causing chronic disease would be a mononucleosis. It tends to develop slowly, persist for long periods of time, and then get worse. Hepatitis B, if you don't throw off that infection, it can lead to a chronic infection causing chronic disease. There are diseases in between acute disease and a chronic disease. We're not going to talk about it. It has a term, but we're not going to talk about it. And I'm sure none of you have ever heard of subacute sclerosing panencephalitis. If you had, you can tell me that I'm mistaken, and I have got a student who's heard of it. I know somebody who got it, but that's an example of a disease in between acute disease and a chronic disease. Infections can also be classified according to the extent to which the host body is affected. A local infection is one where the pathogens are limited to a small area of the body. Like a skin boil is a local infection of Staphylococcus aureus. An abscess, also a local infection. The corollary or opposite of that, I guess that's the opposite of that, is a systemic infection. also called a generalized infection, where the infection spreads throughout the body. For example, when a patient comes down with measles, that starts growing usually in the lungs, and then it's carried by the blood to elsewhere in the body, and that is a systemic infection. Once it spreads beyond the lungs. A focal infection is a systemic infection that began as a local infection. So initially you could have MRSA on your skin, and it may not do anything initially, but then it can cause a skin boil, causing the infection. And then the doctor treats it, and it's MRSA, so it's resistant to the treatment. And then it spreads into the blood, and it's carried by the blood to all parts of the body, such as being carried to the skin, or being carried to the bone, or being carried to the liver. And that would be a focal infection. Any question about any of that? When we're talking about bacteria in the blood, if you happen to see bacteria in the blood, that is bacteremia, the presence of bacteria in the blood. If you have the ability to determine if the bacteria is growing in the blood, then we call it septicemia. So the two terms are similar, but they're slightly different. Usually, we and most clinicians do not have the ability to determine whether the bacteria is growing in the blood, in which case we just use the term bacteremia. Toxemia is presence of toxins in the blood. Viremia, presence of viruses in the blood. So the primary infection is usually an acute infection that causes disease in one location. And you can say the primary infection is the initial illness. Once the primary infection occurs, you can have secondary infection. Secondary infection is usually caused by an opportunistic infection after the primary infection has predisposed the individual to getting the secondary infection. What happens is that the primary infection weakens the host, either weakens the host immune system or does damage to the host. And then the normal microbiota can then become opportunistic as it grows in a location it could not grow in before the primary infection damaged the region. And a good example of this would be the mucous membranes. They can protect us from some diseases, assuming your mucous membranes are intact. They simply don't allow the bacteria or other microbe through the lining of the mucous membrane. But if that lining is damaged by another organism that we call the primary infection, then the opportunistic pathogen can grow. Any questions about any of that? A patient can have a disease-causing organisms and be subclinical. Subclinical disease is one where there's no noticeable signs and symptoms. Another term for that is asymptomatic disease. If you are subclinical or asymptomatic, that does not mean that you cannot spread disease. And I'm using two negatives there. So let me word it more clearly. And that is, if you're asymptomatic, you can spread disease. For example, a lot of young people get infected with COVID-19. But because of they're young and COVID-19 doesn't tend to hit the young, they can be asymptomatic. Your immune system can control the disease and keep it from being symptomatic, meaning they keep it to being asymptomatic. Any questions about subclinical disease? Another example of a subclinical disease is some people can carry the polio virus and not come down with the disease polio. And because of this, it actually will be hard to get rid of the polio virus using the vaccine. It's just that some people can be asymptomatic. We'll talk a little bit more about the polio virus when we talk about viruses later in the term. A little more on the severity or duration of disease. A latent disease is a disease where the patient has no signs or symptoms. If the patient had disease and had signs or symptoms, we then call it latency, where the patient has no signs or symptoms. That doesn't mean that they have thrown off the disease. They still have the disease, and the disease can come back to them. A good example of a latent disease is chickenpox, which usually infects a young individual. Doesn't always, but most of the time it infects young people. And the disease isn't very serious when chickenpox hits a child or a young individual. And then years later, maybe decade later, the patient can get chickenpox again. In between, not chickenpox, sorry, can get the chickenpox virus causing shingles. The first time you get the disease is called chickenpox. All later times you get the disease, it's called shingles. And yes, those two diseases have different signs and symptoms. We'll talk a little more about that when we talk about viruses. What was the point I was getting in here? Oh, a good example of a latent disease is the chickenpox. And usually a child gets the disease, and then the disease runs its course. The individual still contains the chickenpox virus, and about 15% of them will develop shingles. Or is it 25%? Something like that. I'll tell you exactly when we're talking about viruses. But the point is that the virus causing chickenpox and the shingles can go latent, and it can remain in latency for decades. And usually what happens is when somebody becomes elderly, the chickenpox virus can come back and it will cause shingles. The point is simply that the virus causing chickenpox is a very good example of a latent disease, because there's a very long timeline in between getting chickenpox and getting shingles. Another latent disease would be a cold sore. The first time you get it, it's a cold sore. All other later times, it's still a cold sore. But once you've had a cold sore in the future, you can get another cold sore, because the virus can go latent, and then when it's not latent, the virus can return and then go and cause a disease. In this case, we're calling it a cold sore. Any questions about a latent disease? If not, let's talk about herd immunity and then I'll probably end. Herd immunity is when many immune individuals are in the population. It's vital to have herd immunity, and that way that prevents the spread of an infectious disease. Let's see if I can blow this up. So here we have a population, and most of the people are, actually all of the people are not immune. And this would be like COVID-19 initially, when nobody had it. And whenever one person became infected, they would infect other people around them, who would infect other people around them, until most of the population could have gotten the disease. Obviously, this didn't happen with COVID-19 because we were taking steps to prevent its spread. But this is what would have happened if we would not have done any steps to prevent its spread. Most of the human population would have come down with COVID-19. Now, once you come down with the disease or you receive a vaccine, most of the population will be immune to that disease. And those are the yellow individuals. They have either had this disease before or they're immune to it. Now, when an individual becomes infected, they will only spread the disease to a few people who are in contact with the contagious person. And then much fewer people in the population become infected because most of the individuals are immune. And when the virus leaves this infected person, it can't grow in the immune people. And so it protects the unimmune people in the population because they're rare in number. And this we get like with the measles every once in a while. Measles will happen in an individual, and there's enough children in particular around that are immune to the measles, so the disease does not tend to spread. Now, obviously, every once in a while, when the immunity drops too low, the disease can then spread. There was a case where people weren't getting their children the measles vaccine, and they had a whole bunch of children in a Disneyland or Disney World, one of the two. And one of the child came down with the measles, and then they spread it to a large number of other children at Disneyland or Disney World because very few percentage of the children were immunized. For some diseases like measles and COVID-19, you need a very, gosh, I can't seem to get that. There we go. You need a very high number of the people to be immune before we get herd immunity. With COVID-19, they estimate that at least 85% of the population has to become immune, either through vaccination or for prior exposure, before the virus will stop spreading. And of course, we don't have 85% of the population with the vaccine, or 85% of the population who have been previously exposed to COVID-19. And another problem with COVID-19 is a new variant comes out, and then the previous immunity will no longer work. And that will be a problem with COVID-19. And we'll talk a little bit more about that in the future. But they estimate that we'll have to get a booster about every 6 months, or at least every year. to prevent the spread of COVID-19. With some other diseases, you do not need to have a very high immunization rate. And I can't think of a case of it right now, but well, tuberculosis would be a good one, but in the United States, we don't tend to immunize people to tuberculosis. So I can't think of a good example of a disease where it's easier to get the population immunized. What's the disease where the vaccination has gotten rid of the disease? Smallpox would be an example where we had enough of the population immunized, and then we actually made the disease smallpox go extinct in the human population because it wasn't able to spread from one patient to another. And then it eventually died out in the person who was infected. And there's no longer the spread of smallpox. Any question about any of that? All right, if there's no questions, I'm going to end it here. And I will see you next time, unless you got questions, in which case I will be in the lab for about 15 minutes. All right, I'll see you later.